It's a pleasure to be here. It was an exciting morning already, I thought, with a great introduction into signal processing by Mitya Szlowski and questions about how the brain may approach this. And you've seen a lot of functional recordings this morning. Tuning curves, electrical signals, action potentials, firing rates. What I want to introduce or speak about is, if you want, the other side of what the brain is, which is a structural phenomenon. So this this session will be about the brain as a network and how we are trying to find out about it. And I hope it will become evident why maybe it is a very nicely complementary tool to the kind of data that you've seen this morning. As Partha introduced, I'm in Munich now with my group at the Max Planck Institute. A lot of the work I'm going to show comes from the Heidelberg Max Planck Institute, where I was working before as a postdoc back then with Winfried Denk, just to attribute the data. Now, what is it that we think is so interesting about our brains? And each of you, we all have what is called a human brain, and what we know about it, I'm sorry, I'm just looking for a laser pointer. No, I'm assuming we are all of that species. Okay, so one stunning number that is pretty recent and pretty well established now is that all of us have approximately 85 billion neurons in our brains. The SD that the study reports is 5 billion, so we don't know what that means, but 85 billion is the number. So that is stunning. You could say that that alone makes up for some quite exciting organ. Now, somebody could come and say, well, your livers have also many cells, several billion hepatocytes as far as we know. And so that is not alone a very amazing or impressive number. However, what is really a difference between the liver and our brains is that a given liver cell has only a few nearest neighbors, in the sense that it directly interacts with these other cells, mainly those directly surrounding it. While as far as we know today, each neuron has on the order of 1,000 other neurons that it directly synaptically couples to. That is the number. It comes from mouse, but it's probably roughly also true in man. So that is the order of magnitude we are talking about. We have a graph in the human brain of 85 million nodes, if you want, but the order of it is roughly 1,000. That is really different to any basically other organ that we have. Yeah.
That doesn't include the number of glial cells, right?
No, this is purely the number of neurons. And actually, yeah, one can ask, that's actually an interesting question, so where are most of these neurons? Let's say all of us have 85 billion each. Where are most of these neurons? What do you think? Yeah, okay, that's exactly. So what is called a small brain, at least in some languages, the cerebellum is actually the part with the largest fraction of neurons, roughly 2/3, and then the rest is distributed around other parts of the brain. And cortex is not actually the biggest, right? So it's really amazing where all these cells are. Yeah, so glial cells, one thinks today, is another 100 billion or so in humans. Okay, so that argument alone, and that is very simple and very first order, but in a way, this argument alone can, I think, make the point why, if you want to understand the brain, it's very unlikely that you will do so without knowing something about this graph structure. Just because this is what makes it special. It's a little bit difficult argument, but I think it's one that kind of at least can motivate why it's interesting to look at it. Now, before getting into the details, I want to just spend a few minutes on speculating a little bit why this is really promising or not promising, especially because there has been a lot of debate about this. And this is roughly what the talk will be like. It will be first methodological about what we're doing, and then data, and then we'll have a more like practical part at the end also. So one thing that has been often mentioned is that, of course, the argument that structure predicts function has been reasonably successful in structural biology, where often only the crystal structure has really allowed to distinguish or to decide which of the many models that had been proposed kinetically or biochemically is actually true. And examples are the ribosome or the potassium channel with the gating mechanism. So this is just an analogy argument why structural studies are helpful even if you're interested in function. And this is why we like the term structural neurobiology for what we are doing. There are other terms like connectomics that sound a little bit more fancy, but I think structural neurobiology makes the point that this is a comparable endeavor like it was in structural biology. Now, there is a strong argument to be made why, or a question to be posed, why is it so important to look at structural properties of circuits? Again, this morning you saw a lot of functional data, and that is very insightful. But I want to take you through one argument that we've made, which I think nicely illustrates where the limits are. So let's assume we have some black box computation between an input of some odd number of neurons and some very simple output. Let's assume all is binary. Now, how do we find out about the transformation between these inputs and these outputs? One option to do that is to do functional recordings, which, however, amount to a very large number of functional recordings if you want to sample the entire input space. So already in a space of 16 binary units, that is a lot of experiments, and it's much larger, of course, in most realistic settings. And this input-output table just basically lists all the possible input combinations and lists the binary outputs. And now you could have a case where the table is very, very simple, kind of has a very regular structure, and there's only one exception. This is something that is very difficult to find out functionally. However, this could correspond structurally to a case where the largest fraction of the table is is it understandable? Yeah. It's generated by a very simple circuit, and then only for a very, very small subset of the inputs something else happens. So let me take you through this again. So basically the idea is you have some inputs, you have a binary output. You could have a circuit that is very stereotypic here that generates this very, very kind of regular output pattern in the very largest fraction of the entire input space. But then there's this one exception, which is if all inputs are 1, then you kind of flip the output. That would be 1 out of many, many items in the functional table. So if you were to do functional recordings, in the worst case, you have to do all of these measurements just to find out this particular deviation from some generality or, that you have found. However, if you do a structural measurement, the argument, at least that's how we try to make it, if you do a structural argument, you are able to, you can discover such circuits elements by not testing all possible kinds of circuits that you could find, but by measuring the one that is implemented. So, and this is actually really, I think, a reasonably strong point to be made, and I will try to substantiate that later with some examples from our own data, that the space of possible graphs, if you want, of possible connectivity diagrams is so large that measuring one that is actually implemented cuts down hypothesis space about how things are working massively, and much more so than a single functional experiment and even a substantial fraction of the functional experiments. So this is an argument for structural work. There's another argument that is sometimes made to deter us from what we are doing, and that comes from genomics. So all of you know genomics, and it's been a massive endeavor in the end of the last century leading to the sequencing of the human genome. And some people say, well, how is connectomics, what we are trying to do, any better than genomics? It's just going to take up a lot of money, and then in the end you have some data, and it didn't really help a lot. Now, first of all, one can question whether the human genome helped or not. In the medical world, this is, of course, very helpful because you can try to understand the location of, or the non-location of certain disease-related genes. That is not really localized, but you can understand the distribution of them. But even, I think there's another point to be made. So I think the argument is that the genetic code, and I know that this is not how the genetic code looks, actually is 2 steps away from function, because you first have to translate the genome into something 3-dimensional, and then you get basically the functional result. In the case of neuronal circuits, however, I think you could argue that the circuit is really more comparable to the 3D structure of proteins, and that it is much closer to to generating the function that you're interested in. So in a way, therefore, we have a certain advantage over the linear sequencing in genomics because we're closer to the functional output of the system. Again, there are points to be made against that argument. I'm happy to discuss that. But I just wanted to give you a little bit of arguments that occur currently a lot, and maybe some ideas of how one may deal with that. Okay, so this is about why circuits are interesting and a little bit in what context we are. Maybe are there any counter arguments so far, or do you want to get into this debate, or should we continue? So any questions so far or arguments against what I said? Okay, I'm pretty sure this will happen later. Good. So now how can we do it? Let's say we agree even that we want circuits. How do we do it? And the most common way of mapping circuits, at least in, for instance, the cortex of mice or so far has been to use electrodes. Let's take a setting in cortex of a mouse. This is a rat, or rat in this case, somatosensory cortex. You have 4,000 cells in what is called a barrel. And this is actually an actual map of all of these cells. And what people do is they take 2 electrodes at a time and stick them in and ask whether 2 neurons are connected synaptically, functionally measured. This is a very important experiment because it leads you to a very representative number of a circuit, which is the average pairwise connectivity in that circuit, which turned out to be 15% in that circuit. And that is actually a number that the more we know about local circuits, the more it seems to be true. This is roughly the local connectivity in most local circuits. That means if you have a neuron, then of those neurons that are reasonably close, roughly 15% or maybe 10 to 20% are directly synaptically connected. So that's kind of the local circuit density, and then it gets much, much sparser, of course, the more you're going at larger distances. Okay, that's an important number, but it's not all, as you can see in the sketch here. So what this is supposed to be is a graph of only 100 neurons. And I've just compiled 3 different kinds of circuits that you could potentially think of. The first one is shown here in what you can call a connectivity matrix, or this is just generally an adjacency matrix of the graph here, where the white part is 0 and the blue dots are supposed to be 1, which means synaptically connected. And then you can plot it like this, where you have a presynaptic and a postsynaptic side. And again, everything that's blue is connected, and the rest is not. Now, you can imagine this to be really just pairwise random. And that is what has been coined Peter's rule for historical reasons in anatomy, which means that all there is pairwise connectivity statistics. That is, all you can know about this matrix would be that it's filled with 15% filling fraction. Okay, so 15% of those entries are non-zero. Now, however, you could think of a completely different kind of circuit, which would be this very simple example of 2 cell assemblies that are highly connected amongst each other, but very sparsely so between each other. As shown here in red and blue, could look like this. Now, the point is that the filling fraction of this matrix is also 15%, which means that by just measuring pairwise connectivity statistics, you have no chance to distinguish between that circuit model and that circuit model. And even more so, or as well, for models like Sinfire chains, which could also occur, or have been proposed at least, and could, of course, also generate a 15% average pairwise connectivity. So the point is that as long as you're just sampling a given circuit once for one pair of connections, you have no chance to distinguish such circuit models. There has been a very interesting recent approach using viruses that travel across synapses, and this is called transsynaptic tracing. This is a big advance because it allows you to sample an entire column in the connectivity matrix at once. Still, if you don't use multiple colors and don't do 2 experiments of this kind in the same circuit, it's still not possible to distinguish those 2 models. Right? If you only can do one of these experiments in one circuit and then you have to take another circuit for the other measurement, then this comparison is still not possible. So really what you need is a complete mapping of such matrices to find out about circuit structure like this. Okay, when I say complete, I mean sufficiently complete or reasonably complete. So of course, you can argue it is possible to to sample. Given the type of circuit you're looking for, it is possible to sample the circuit if you know what you're doing, but pairwise sampling clearly is not sufficient to distinguish these models. Okay, and that is why people have tried to reconstruct neuronal circuits. Now, you could say, okay, that sounds reasonably plausible. Why hasn't that been done decades ago? And people, or scientists have worked on that, of course. The problem is that neurons are very difficult to reconstruct for two reasons. And the one is that they are very small. So neurons at their thinnest are easily 40 or 50 maybe nanometers. Some are actually even much thinner. And at the same time, they are pretty large on that scale. That is easily hundreds of microns is what they span, if not millimeters. And this combination of very small and very large makes it very difficult to reconstruct neurons and many neurons in their entirety. So let's briefly visit these numbers. Where do you think are the thinnest parts of neurons? So 50 nanometers, something like that. Where does it occur? Where do neurons get so thin? It could be, yeah, dendrite, but which part of the dendrite? Yeah, okay, so the picture, everybody has his or her own picture of a neuron, which is usually based on the first textbook you read. Now, if it was one that started with peripheral neuroscience, then you would probably have the picture of a or pseudo-unipolar neuron, which is the one that comes from the sensor and goes through the spinal cord up to the brain. This is then your picture of a neuron with some receiving part, a long transmitting part, and then usually one synapse at the end, something like that. That is a common picture, that you see a synapse like this, and one is led to think that this is the one end of a neuron. Okay, that's one picture you can have. Now, if yeah, there are other pictures, but I want to propose one that is true for most cells that, not most cells, but many cells that you have. And that is that of a pyramidal neuron. Who knows a pyramidal neuron? Okay, so it exists both in cortex and in hippocampus, and it has roughly this kind of picture where you have a cell body, and you have dendrites, which are these here. This is an apical dendrite, and it has this weird shape. Then it has a kind of triangular shape down here, and you have basal dendrites. So that's all dendrites. And then you have one axon going out. But this axon very quickly branches massively, and oftentimes actually in these weird, almost perpendicular ways. And this branching here is actually what really makes up most of the cell. So this is a pyramidal neuron. So dendrite here, axon here, and most of the synapses, one should say, are those en passant synapses that happen along this axon. So this picture of an end synapse is very rare, that the axon ends and makes a synapse. In most cases, the axon makes synapses all the way. Okay, so this is an attempt to reshape or reinforce the picture of a neuron. Now, why is this cell so interesting? Who knows where these exist? The pyramidal cells, pyramidal cortex. Yeah. So in our cortices, and what fraction of cells in the cortex are pyramidal cells? Roughly. Many? Yeah, the absolute majority. So the number is roughly 85% of your neurons in your cortex. I can't say in your brain because the cerebellum takes up most of the neurons, but in your cortex, this is where really the cool stuff happens, which is detection of stuff and probably thinking and all that, these kind of things. 85% of the neurons up there are those kinds of neurons. Very stereotypical. It's amazing how stereotypical they are. So far, they're really treated as a kind of still almost one type of neurons, although that's probably not true. So this is the neuron. So back to the question. The dendrites are actually, and that's the way I drew it, are the larger, are the wider, the bigger parts of a cell, in the sense that the diameter is more on the scale of a micrometer, at least roughly speaking. While the axon actually gets very thin and can get smaller than 100 nanometers. Typically, it's more like half a micron or so in diameter. So this already is one of the answers to where a neuron can get very thin, which is thin parts of axons. But I have to be fair and say that these neurons have what is called spines, which are small protrusions on the dendrite. So if you magnify it, it looks like this. So like thorns on the dendrite, which have a very thin neck and then a head. That is at least how it's usually described. And this is what is called a dendritic spine. It's very, very frequent in all of these cool pyramidal neurons in your cortex, also in other cells in the cerebellum, for instance, and also in the hippocampus. So spines are typically the locations where synapses are made. So axons come in and make a bouton here, a synapse here onto the spine, and then that's how the signal travels to the soma. So, and these spines can get very, very thin. So this is one second typical answer to this question is it's the thin axons and it's the spine necks that are very, very thin. And then, of course, you have animals like flies where almost the entire brain is optimized for being light, right? Because you want to fly around with your brain. And that makes some of the neurites extremely thin. So even typical dendrites in flies can be like 30 nanometers, as people like Mitya have kind of seen now in their beautiful reconstructions that you saw today. So some of the neurites that you saw today that you showed in this nice reconstruction are really thin, like 30, 40 nanometers. So really challenging job. Okay, so the point of all of this was to kind of make clear that neurons can be very small and they are very large, unfortunately. And that means we have to have high resolution over large distances. And this is something that resolution-wise, for the longest time, you could only achieve with electron microscopy. Now, some of you may know there are recent developments of super resolution light microscopy techniques. I'm happy to discuss the hopes and benefits of that, but basically one has to say 50 nanometer and less is basically still the realm of electron-based optics. Okay, so how can this be done? And you've seen some images today. So the method used ever since electron microscopy was introduced into biology in the 50s and 60s of the last century by Colonier and Gray and these people is to take a block of tissue and then slice up your tissue into very, very thin sections, typically 50 nanometers or at the time 90 nanometers, and then put the tissue section into a transmission electron microscope where you shoot an electron beam through the sample, if you want, and you detect the deflection of the electrons on the other side. So that is transmission TEM, transmission electron microscopy, and that works very well. Afterwards, you take those images and align them, and then you get a kind of volume sequence. The challenge, however, with that method is that you have to actually physically transfer those sections into the electron microscope. Right? You have to first cut them and then get them into the EM. And sections that are 50 nanometers thin are notoriously difficult to handle, so that is a problem. But even worse so, because in order to get to any reasonable volume, that is a z-axis length of, say, 500 microns or whatever you want, we are talking about tens of thousands of sections. So you do not only have to be able to do that a few times, but tens of thousands of times in a row, or at least thousands of times in a row, to properly transfer this into the EM. And this has really been the main challenge of this method, which is why it has successfully been done only in very few settings. One of them, the famous reconstruction of the C. elegans connectome, I will briefly show that in a second. But this is really so tedious that it was really limited to a few studies only for larger volumes. And therefore, a new technique that Winfried Denk introduced in Heidelberg and that we've been working on over the last 6 or 7 years is what is called a serial block phase scanning electron microscope. Used to have the unpronounceable abbreviation SBF-SEM. We've now converted to SPAM. Not my decision. I mean. Okay, what's the trick? The trick is instead of first cutting a section and then taking an image like people did with the TEM, we invert that process now. And we do that by putting the entire block of tissue into the electron chamber. And then we first take a picture from the top of the block. And then we have an oscillating diamond knife installed into the chamber of the electron microscope that then shapes off or scrapes off, if you want, the top of the block. And then we can take another image and then abrase, image, abrase. And that simple inversion of not first cutting and then imaging, but first imaging and then cutting means that all the images that we take are intrinsically very well aligned because they come still from the moment where the tissue was intact, right? And then only afterwards the physical distortion happens. Yeah.
Sorry. Do you shoot the electrons through the tissue?
Very good question. That's the second difference that we had to then accept, which is, of course, we cannot anymore shoot the electrons through the entire block. That's too much of too long. Therefore, we are using scanning electron microscopy. So instead, we're detecting the backscattered, currently backscattered, but potentially also secondary electrons that come back from the surface. From the surface top, right? So it's either the electrons we shoot in that get deflected all the way back into the detector, which sits at the same location like the emitter, or those that we basically shoot out of the material. So these are the kinds of electrons you detect. Yeah, again, there's a scanning electron microscopy where you scan the surface and record the contrast at each voxel. Okay, and this method now allows us to actually do these experiments over and over again. First of all, because we don't have to carry the section anywhere anymore. We can go very thin, currently typically 25 nanometers section thickness. And also because it's fully automated, we can really run for thousands of slices. And this is what also is shown in this movie here, nice illustration of the process. So you can see the yellow electron beam. So just a mock-up that scans the surface, the image is taken, then the knife comes and scrapes off the top of the block. And yeah, I want you to also notice the way that the knife takes away the tissue. This took a long time to get for the illustration because maybe next time you can see that it nicely curls, you know, and actually it's partly what you spend your time on. Yeah, see how it curls. Good. Okay.
This already happens.
Yeah. Okay. I could say something like it's pretty dark in there, so we don't know. No. Actually, yes, it's not as curly. No, it's more like sometimes the entire slice just happens to stay on the knife, but then sometimes it breaks a little bit. So you're right. So I mean, it does crumble when we take it off, but that is the material that we have imaged already. So yes, so sometimes it slips and then slips on top of each other or so, but that doesn't matter anymore because we've taken the data already. So yeah, you're right. So it's actually. Actually not. So I mean that is one of the challenges actually of the method is to get the knife settings in a way that it really is very smooth on the block, and that is what many parameters go into, but it works. And then what I was referring to is more that the tissue then gets on top of the knife, and then may, for instance, get thinner here and leak, and then the other part will come on top also. That is not, as you will see later, for the imaged surface, that is not a problem. So that is really referring to the problem of the debris, as we call it, which is what is gone. Okay, and the resulting data, at least a while ago, looked like this, where you have an XY image, but if you then take a reslice along Z, then just at first sight, the images look very similar, which gives you an indication of the level of isotropy of this method. So again, this direction here is the cutting direction. So this corresponds to many hundreds of image slices. Maybe just for orientation, what do you think you're looking at? What do you think these large blobs are? These large gray things. There's a scale bar. This is somewhere in, not your, but in this case back then, a rabbit's brain, but where? It is brain, or at least what people consider brain. Okay, these large things, cell bodies. Okay, that's just the way they're stained. They look reasonably homogeneous. Where do you have a setting where cell bodies are very much ordered like this? Do you, anybody an idea where this could be? where in the brain. So you have this layering of cell bodies. You have another layer of cell bodies. You have some stuff in between, which is actually the most interesting part, which is the synaptic network. So yeah, okay. So this is a part of Any suggestions? It could be cortex, but actually in cortex cell bodies are much more scrambled. So nowhere. Yeah, but still there, the cell bodies are really like scattered around. There wouldn't be such a layering. Okay, I just want to introduce you to the to your or comparable brains at that level of detail. So yeah, so it's highly packed. So usually people suggest hippocampus. There's also a strong layering. Then the argument is, well, 10 microns for a cell body in hippocampus is a little bit too small, and so on. It gets a little bit more difficult. Actually, what it is retina. So this morning we've heard about the retina, and for the rest of the talk you will hear more about the retina, because that's one of the circuits we are studying intensely. And this is rabbit retina, where these are the ganglion cell bodies of those ganglion cells that you've heard about a lot today. These are cell bodies of amacrine cells, and going further you would have the bipolar cell bodies. And this is actually the layer here in the middle. All the scrambled stuff, if you want, is called the inner plexiform layer. And this is where the synapses between bipolar, amacrine, and ganglion cells happen. So this is basically where the processing between the bipolar cell signals and the ganglion cell output happens. That's all in here, within these like 30 microns or so. We'll see more of that. For so far, just the methodological point that the reslice is very homogeneous. Let me briefly step back and just make sure that I've mentioned all the possible approaches to EM-based circuit reconstruction. I mentioned already the serial section TEM, and I will show you a little bit of that in a second again. There is another very recent development of a serial section EM, but actually imaging the sections with scanning EM, where the process of collecting the sections is replaced by a conveyor belt mechanism that kind of automatically picks up the sections. This is a method developed by Jeff Lichtman's group at Harvard, which they call ATOM or ATLUM, depending on the details of the method. This is potentially also very high throughput, because this automated section collection can get very reliable. There's a third method that uses, instead of the diamond knife, a focused ion beam to mill away the top of the block. We'll briefly show something there, and then the method that we're using I already showed. Yes?
The parts that you got from the SPEM,
Mm-hmm.
Can they be used, or they kind of like that you cannot use them?
No, so the question is whether we, in our method, can still use what we've cut away, and the answer is no. So we really have, our method is what you can call a highly destructive method. So
Okay, because then you cannot then compare to the SSTEM.
That's correct.
If you can have like the same result.
Well, I mean, okay, so it is true that in the serial section TEM and also in the ATLUM method, you can potentially, or you can retain your sections. You have them physically there. The question is, what do you do with them? And the comparison we are doing is that for circuit reconstruction. And for circuit reconstruction, you want a volume of reasonable size, because you want to follow all the neurons, and you want resolution, and you want a good alignment. So these are the criteria under which we evaluate it. Revisiting your data is also, or your slice is also something that may be useful for certain settings, like immuno or so. But for the circuit reconstruction, actually, we think it's not that important.
How do you compare the focused ion beam and the diamond knife method?
Yeah. So can I get back to this in a minute? I have data there for that. So just briefly to mention the most famous example of a serial section TEM reconstruction, I want to show these slides from the C. elegans wiring diagram reconstruction. This was done by Sidney Brenner and colleagues in the late 70s and 80s. Who has heard of this, of the C. elegans connectome? Yeah, okay, so this is well known for good reasons, because it was a big achievement at the time. It took them 15 years, roughly. And it resulted in this beautiful publication in the Philosophical Transactions. It's a 340-page paper. For those who haven't seen it, I really recommend having a look at it, because it has a almost aesthetic value, not just for the size, and the beauty of the data, but also for the running title. So the running title is that is on the second pages is "The Mind of a Worm," and I find that is kind of pretty a nice running title, given that usually it's like whatever enzyme does whatever, the mind of a worm. Okay, so that is the reconstruction they did, and Mitya Shklovsky and collaborators have revisited this data, and over the last 5 years published reanalysis and also some, one has to say, corrections or additions to that wiring diagram that were based on the data that Brenner and colleagues had collected. So this is an example of revisiting, actually, where the data was still there and the pictures were still there, at least, and they could revisit the data. And this is last year they published the final result for this data set, where this is, if you want, the connectivity matrix they came up with for C. elegans, where I think red is chemical and blue is gap junctional, that is electrical synapses. Okay, so this is C. elegans, 302 neurons, each neuron less than a millimeter in path length, a very small organism, but all of the neurons, at least for the hermaphrodite, very nice job, but once in 15 years. By the way, it was 9,000 sections, I think, from 3 animals that they used. Okay, FIBSAM that Partha asked about. So here the method is to, instead of using a diamond knife, to use a focused ion beam to get rid of the top of the block. So you have basically two, if you're shooting 2 things at the tissue if you want. One is electrons to image, and the other is mostly gallium ions to abrase the surface. So you get such a setting where you kind of shoot 2 things at the tissue. And that data can look very nice, actually. I will show you a movie from one of their publications. This is an example taken, I think, from cortex using focused ion beam imaging, and you can see that the resolution is very high. You can see vesicles, and this is a myelinated axon, and so it's a very kind of precise method to image. So each of the synaptic vesicles really is very clearly visible. That is very helpful. So that is a clear pro. The resolution is very high. The limits of this method are volume. So currently, actually, everybody agrees that it's a limit to something like 20 microns on a site. Now, that is kind of the current state of, or that at least that is the published state, if you want. There are novel developments, some of them happening in Genelia, to try to extend the volume of FIB milling or FIB-based imaging to hundreds of microns. But yeah, so I mean, that is of course an exciting route. Currently, one I would usually the way I put it is for small volumes, and small being like 20 microns on the side, roughly. This is the optimal technique because it's really very high resolution and gives you very, very precise data. Okay, so this was focused ion beam milling, and yeah, I guess that should serve as an overview. I showed you a little bit about SST and the data that came from it. I showed you an example of FIBSAM. I introduced our method, and I mentioned the ATLUM that has been, at least as a method, been out there, and yeah, it's being used right now. Okay, so let's stick with methods a little bit and talk about reconstruction of this data. From the beginning on, people tried to use computers to help them with reconstructing this data. I just thought it was nice to see that already in '72, the computer was suggested as a tool to help reconstructing this data. And then there was a phase where reconstruction was done using on these acetate sheets, and then you would get reconstructions looking like this, where you can nicely see the kind of single slices from which the data was assembled into a 3D reconstruction of a piece of a neuron. That's, I think, from Sterling's work. And then another tool was developed by Fiala and collaborators called Reconstruct, which is a software that you can still download and is still being used by many labs, which for reasonably small volumes was a very nice tool to label neurites and put them into 3D reconstructions like is shown here. So these are cases, and by the way, this is actually a dendrite with spines, so the one that we talked about from, in this case, I guess, hippocampus, where you have the dendrites, and then you can see these little protrusions, which are the spines. So this is reconstruct and has been excessively used. A more recent development is are tools that are integrated into the Fiji development, which is basically the successor of ImageJ, as you may know, and then there's especially Tracky-M, and then there's a development from, again, Janelia and Anjan Peng's group, which is called V3D. It's also published, that allows you to integrate many kinds of data, including neuronal reconstructions, but also atlas forming data that comes from light microscopy and is made basically for their efforts of creating fly brain atlases. To get an idea of how tedious reconstruction can be, this is an example using some MATLAB-based tool that we have written just for our own purposes, where you can zoom into the data and then outline neurites. But as you can see, this is pretty slow if you want to do it accurately. You can do that in all dimensions if you want, but just to give an idea of how slow this can be, if you want to be reasonably precise. And if you do that for a while and spend some time on it, then you can take data like this, which is like 2 or 3 years old data from the method I introduced, the SPIFSAM method, SPAM method. And this is retina again, where a cell is reconstructed. You can see the tissue block as a whole, but then the cell, one of the neurons in that tissue block is coming out. This is actually an amacrine cell, which has been reconstructed in here. And that's, of course, nice to look at, and you can reconstruct more neurons. But in terms of methods, this is just incredibly tedious. So if you look at the speed of such a reconstruction, the numbers we and others have to spend are roughly a few hundred hours per millimeter path length of a neuron. Now, what is that unit of millimeter path length? This is how we like to describe the challenge, because this is not about the distance you travel. This is really the path length of the entire graph you're reconstructing, right? Or of the, say, of the neuron tree that you're reconstructing. And how does that translate into the real world? Well, most circuits are large. And let me take 2 examples. One is layer 4 in barrel cortex. If you take the entire layer 4 of one barrel, this is roughly 4,000 neurons in a rat. This is a part of the cortex that represents sensory stimuli. You can talk about it if you want. There you can add it up and you get to something like 40-ish meters of path length. Even the mouse, a local piece of mouse, is easily half a meter or a meter in path length. So this is the total path length of a circuit, and in most parts of the brain that you may be interested in, it's easily meters. Okay? That means that a reconstruction speed of the kind I just reported is reasonably prohibitive, right? We're talking about millions of work hours, which, I mean, even if you have really, really dedicated people in your lab, is very, very difficult to get to. And this means that really, for the longest time, with these kinds of reconstruction speeds, substantial circuit reconstruction was not thinkable, because that's really even beyond massive funding scales. Okay. Therefore, one thing we did to be able to handle our data is develop tools to, first of all, browse data, but also annotate it more efficiently. And this is what I'm showing here, which is the tool called Knossos, which first of all allowed us to 3D navigate through this kind of EM data. You have 3 planes which depict the orthogonal views of the data, and we made it such that you don't have to store the data sets in memory, because they're large, terabytes large. So you can run it on a laptop, and that's a nice thing, because then you can actually distribute it to many people. But then also you can annotate in a way where you only annotate the center line of a neurite. Okay? So instead of contouring each section, you just basically fly along the neurite for a while and drop another point while doing so. And this is, as you can imagine, much more efficient than the contouring process we had seen before. And just to get a more visual impression, again, this is a movie that now flies along such an axon. So this is a bipolar cell axon in the retina. We're flying along it into the retina right now, into the inner plexiform layer. Whenever there's a branch point, we have this dot as an indicator, and the plane of the flight is almost or supposed to be orthogonal to the center line of the neurite. I want you to focus on two things. One is that if you just watch the neurite and how it flies along, you can see that it's possible to follow it, but also you may see when it rewinds now for a little bit that actually the data around it is very, very complex. Right? So there's a lot happening. In other words, your brains are really, really densely packed with neurites. Right? It's not like there's one neuron and then there's a little bit of free space and then there comes maybe another one. Like, again, most of the movies about the brain look like that, right? Because you can't depict it otherwise. But actually, this is how dense, roughly speaking, how dense your brains are.
So a quick question. So having reconstructed for example, something like the center of gravity of the conceptual
Mm-hmm.
can you use machine vision to reconstruct the contour?
Yeah, that's actually what we're doing to kind of get a little bit further. It's still not optimal because, of course, ideally we would like to turn everything to computer work, but that's exactly what we're doing. I'll show that. Okay, but that was to show this idea of flying through your brains, which is something we'll pick up later. Okay, so what did this gain? In our hands, it gave us a 50-time speed up, which was reasonably substantial. It's still slow, so we're talking still about hours per millimeter path length, but this speed up, as I will show later, allowed us to actually go for sizable circuits, which wouldn't have been possible otherwise. Now, of course, in such settings, it's always a trade-off between speed and accuracy. So let me briefly, and that is the last methodological remark, let me briefly talk about the accuracy of these kinds of reconstructions and how we try to compensate for that. So when we wondered whether things can be nicely reconstructed, we asked two, if you want, self-proclaimed experts to reconstruct the same neuron. And this is what we got. So we got a reconstruction where in green is agreement between those two so-called experts, and then in red it was disagreement. And there were 13 or so locations at which the two disagreed, and that was reasonably puzzling because, yeah, as I said, experts, and more puzzlingly so, this was my close collaborator Kevin Brigman and myself. So we didn't like this point, and then we revisited the locations where there were differences. And luckily we found that most of these were actually very easy to reconcile. So that was weird. So one example is here. It's difficult to show in 2D, but it was basically a very clear branch that one of us had missed. And what we learned from this experiment, but also then from more massive analysis, is that actually one big challenge in this kind of reconstruction is attention. So, and that is because the data is so massive, right? So if you travel along one neurite, you have to be constantly attentive not to miss a branch going off. That is difficult from a single neuron reconstruction where you have stained just the neuron, where you know that everything that's green or black or whatever is that neuron, right? So the bias usually is much stronger that any signal you see belongs to a neuron. Here, we have neurons everywhere, and it's absolutely not clear that any of the other pieces that we see belong to the same neuron that we're trying to reconstruct. So this means that even experts make errors. We can't rely on single annotations. We have to have some way of correcting for these kinds of errors. Yeah, again, attention related, may have to do something with caffeine uptake or something like that, but even, yeah, I mean, most of the errors were made luckily by Kevin, but I had more coffee, but then it doesn't scale and so on. So it's really, it doesn't work. So we can't just rely on single humans. Well, what do you do to overcome such a setting? And that is not the only example, there are other examples, is multi-annotator evaluation. That's what we do. So we ask many people to start at the same location. Now is the purple tracer. And the idea is that if the errors that they make are reasonably uncorrelated because they are attention related, then using their combined annotations, I'll get back to your question in a second, using their combined annotations, we can actually find out about what the most likely correct result is. And more specifically, we devised a metric between those linearized neuron reconstructions that allows us for each location along a neurite to just count basically or evaluate the agreement between multiple annotators, and then use this agreement distribution to write down a decision model about how people take decisions here based the data, and then just optimize our decision using those measured priors and this decision model. And then you can come up with rules about whether you reject a certain annotation or you accept it. And then if you do that for an entire neuron, then you can get rid of the stuff that is unlikely to be correct, and then come up with what you can call a consensus. But you had a question.
Draw most of the neurons where branches go out from.
Mhm.
There's one coming in. You have to go down all again, or like going up and down the planes all the time.
So that depends. Okay, I think I understand what you mean. So neurons are reasonably isotropic. So they, except for a few cases, they don't respect certain directions really very strongly. Therefore, you need some reconstruction that allows you to be reasonably variable in direction. And what we did at the time and have done so far mostly is we used the 3 orthogonal views, which is of course in a way a sampling of the directions that are optimally reconstructable. Ideally, you would use a free plane reslice, which is something we're doing right now, which is even easier. Yeah, but you're right. So you have to be able to do that in 3D. And that's what Knossos came closest to at the time. Okay, and then we have a consensus, and this is again a pictorial view. So we asked many people, really many, to reconstruct the same neuron. And this is an example where we asked 50 students to reconstruct the same cell, and this is what we got, which looked pretty messy. And then if we applied these decision algorithms, if you want, then we get what you can call a consensus skeleton, which is, given these annotations, the most likely truth. Okay, so this is very briefly just to give you the idea of how this problem of massive data annotation is currently tackled in the field mostly. And the answer is a combination of, as we see in a second, machine learning, but here crowdsourcing. So really, this is a case where data annotation is so massive that we and others have to turn to crowdsourcing. And that's not the first time in scientific history that happens, right? I mean, there have been other examples. I think in early days of astrophysics, there's still a lot of annotation of galaxies online even. And then maybe some remember that I think in early years of what I wanted to say of particle physics, I think there were people doing tracked trajectory annotations. I think also in that time it were like housewives were recruited to like in the 50s or so to like track particle trajectories. Again, because annotation was just so difficult and so massive. It's a comparable situation here with other clientele. Okay, so. Of course, you may ask yourself all the time, why spend so much effort on humans? Why don't we take computers to do this job? I want to briefly just illustrate what the challenge is. We want to convert gray value data into segmentations. It's evident that this, as a global inference problem, is not really tractable, because if you change something about the segmentation locally, because the neurons are large, it will affect the segmentation at the other part of the tissue block in the worst case. So basically what you do is you do some local classification, apply some local classification of the data, and then try to use some more or less smart segmentation algorithm to propagate the local classifications into segmentations. Now, this is a very general setting that is nice, because then you can apply all the methods for machine learning, right? So basically, the way you can treat it is you can say that the data is some surround of gray values around a given location. That is a high, externally at least a high-dimensional space made up by these gray values. And then you want to classify that into something simple, usually, like I'm in a cell or not, or this is a wall, or this is a membrane, or something like that. So you have a high-dimensional input space, low-dimensional classification space. This is a typical machine learning setting. So we and others have played with machine learning methods to classify this kind of data and reconstruct it. Here's an example from some template matching approach, which basically tries to almost binarize the data and then try to reconstruct it. And yes, you can get very nice reconstructions that look like this, where neurons are nicely reconstructed over quite some distance. This looks nice. However, if you look closer, there are still many errors. Specifically, there's a large fraction of objects in these reconstructions that are not properly assigned. And this was a very strong point to be made, that although at first the prospects looked really nice and people thought that computer analysis can get us quite some distance, because when you look at this it looks nice, that actually because neurons are very long, we're not there. So there's the best algorithms in the field, I don't have that slide here, but are currently at a stage where you can automatically reconstruct maybe 40 microns a piece. Now, a neuron is millimeters long. That means that after a very small fraction of the neuron, there's an error on average at least, which means you lose the largest fraction of the synapses. So it's really a setting where that is apparently difficult enough that just low level, if you want image processing, has not gotten us to performance like humans. And that is true for all the methods that are out there right now. And therefore, we've and others have used a combination of crowdsourcing and machine learning.
Did you combine your two approaches?
Yeah, okay, that's exactly what we're doing. So we're using humans to do the long-range annotation and computers to locally blow up the volumes. So exactly what you basically suggested. And that is in our hands a very kind of valuable approach, because we can focus humans on just getting the long distances right, which is difficult for computers, and we can use the computers to fill up the volumes, which is boring for humans.
But couldn't you also like take the small 40 micron things and ask humans to connect them?
Yeah, very good. Very exactly. That's exactly the next step. So everything I'm going to show you now, the data that I'm going to show you now, is still from the phase where we did whole neuron reconstructions by humans and machine learning to segment on top. What we're doing currently is exactly what you're suggesting. So we're first running a classifier that has a confidence output, tells us where confidence is low, and that is where we're pointing humans.
There's one thing that may be too difficult to do, but in principle one could imagine simultaneously reconstructing all the neurons automatically. Then you get certain constraints.
So yeah, so that's also something we looked at earlier. So the problem is that so there are global constraints. What Path is suggesting, of course, that you know that a neuron has only one cell body. A neuron doesn't have loops, at least as far as we know, in the tree. There are certain branchings that nobody has ever seen, things like that. So there are constraints on this data. Now the problem is that first of all, the first constraints I mentioned are so global that it takes a long time till you get to that scale where that really matters, and that is still beyond what we can do. So this is more like a scale of hundreds of microns at which these kinds of priors would become useful. The second problem is that it's a little bit difficult, because the moment that your reconstruction gets dense enough that you can really use the other neuron to help reconstructing your own neuron is at a very high filling fraction. So basically you have to have already a lot of knowledge before you, I think, in other words, for the last 10 or 20% of the reconstruction, this is roughly where it really helps. For the first 80, it doesn't so much, or not at all, or less so, let's say.
I mean, one thing that we're using is orphan detection.
Yeah.
So when you really have a dense reconstruction, then all the bodies in the volume have to reach outside, because the reconstructed volume is so small. And if you have something that does not connect to anything, that must be a mistake.
Yeah. Yeah. So that is for if there's enough processes that leave the volume, that is something that you can use. It's roughly, again, in our hands, accounting for, this approach is roughly accounting for 30%, maybe 40% of neurites. So a large fraction, not all of them. That do. No. It's more like a third, roughly a third of the processes are coming in and leaving, and not in the retina case. We have a large volume. It's 150 microns or so on the side. So it depends on the volume size, of course, but that is one of the constraints one can use.
So when you say computers can reconstruct only about 40 microns of thing at any given time, I'm wondering if I understood that properly. Can you then restart the process again at the end of 40 microns with a new set of parameters which better define that environment? That is an adaptive tracing algorithm rather than something with fixed parameters.
Yeah. So of course you could think about that. The way the number came from, however, is not the picture where you're starting somewhere and trying to reconstruct and then something happens. It's more like you have a 3D classifier that reconstructs the entire volume, and then you post hoc ask how many errors do you have, and that is more the error rate that I was reporting. And that means that that is already after using all the tricks that people have tried to come up with. Now, this number, of course, is not fixed. I mean, people are working on it. Many groups are working on this and trying to push the boundary further. My impression is strongly that this low level, as you can call it, classification, where you're really just working with the image data and not putting in constraints of the kind mentioned, or using higher level object features or so, seems like bounded around that currently, at least. Okay, but we didn't want to get depressed about automated reconstruction not being too effective at the time, at least, and still with challenges. We wanted to get some data, and that's why we used, as I said, manual reconstruction plus computers to get to results. And I want to illustrate that, and this is the data part, so no more methods, but data, where, and this is not as long, so we'll just, I want to give you some ideas of where we stand in terms of reconstruction. And this is two projects that I want to briefly mention. One is what you can call a sparse or targeted reconstruction. So this was a reasonably large tissue block of the retina, sized 350 times 300 times 60 microns, where we, however, didn't reconstruct all the neurons, but only very few, namely those that we thought were interesting for the particular question that we were looking at. And the other example that will come later is actually a dense reconstruction of a block of tissue. The one is, you can read up, is published. The other is almost. Okay, so what we were looking at were, again, ganglion cells, and were nicely introduced today by Mitya in the morning already. So we were looking for certain types of ganglion cells that respond to directional motion. What is shown in this movie is a way of finding out what kind of response properties these neurons had by functional recordings, not with electrodes, but with functional dyes. So what you see here is blood vessels in the retina. You're looking onto the retina, and all the green stuff is, these are neuronal cell bodies of ganglion cells filled with a calcium indicator, which means that if they flicker a little bit, then you can interpret this as an activation of the neuron. And we mapped those neurons that responded to certain stimulus directions, which are called direction-selective ganglion cells. And then we reconstructed this entire tissue block, the same exact tissue in the electron microscope. So, by the way, this was a series of 13,000 sections, indicating how you can do this very long experiment. And then we were able to map the functionally recorded neurons by their cell bodies onto the EM reconstructed data, and then used this Knossos tool to now follow the processes of that particular ganglion cell into what you see here now, the, again, inner plexiform layer, which is where the ganglion cell receives all the synapses that it then uses to integrate a signal and send it to the LGN, mostly at least, as we heard today. So this now, again, gives you an indication of the scale, I hope. So this is even beyond resolution of the projector, of course. You can't see the single processes anymore if we wouldn't show them for that particular cell. So this is the dendritic arbor of that ganglion cell. If we turn it around, we see that it's a bistratified ganglion cell. That was known that the direction-selective ganglion cells, or one type of them, is bistratified, has two intense projections here. Again, this is the retina in a side view now. We're now again in the planar view. I hope it's not too confusing. And now you can see several of these ganglion cells, and the color here now maps onto their functional response that we had measured in the beginning. Okay, so to recapitulate again, we used functional recordings to identify certain cells that have certain properties, in this case directionality, then took the exact same tissue block into the EM, reconstructed those particular neurons, and wanted to ask what their specific synaptic inputs are that then we hoped would allow us to find out about how, or speculate out, about, or find models about how that directional selectivity is computed. Now, what are the basic ideas? Well, you have excitatory bipolar cell input to ganglion cells, and you have inhibitory amacrine cell input. And more specifically, it was known that a certain type of amacrine cells, the so-called starburst amacrine cell, projects to these direction-selective ganglion cells. And if you want, in a very simple term, the question was, is the directionality of that ganglion cell generated by the excitatory part of the circuit, or more specifically by this inhibitory amacrine cell. And there were strong suggestions that it is actually the inhibitory part. And the reason was that one had found that in the amacrine cell itself, you find directional signals in the following way. So if you stimulate this kind of amacrine cell that you can see here with light stimuli that move outward, you saw stronger calcium responses, or that is excitation, than if the stimulus moved inwards. So that was known for a while. So you had a cell that had a certain directionality detection, and the question is, would the postsynaptic cell inherit it specifically from these presynaptic cells? Okay, so this is the overview of the data. So we had the functional responses of the ganglion cells that allowed us to ask whether those directional inputs are specifically made up by the circuit. For that, we reconstructed not only the ganglion cells, as I had shown you before, but also a large number, or some sizable number of those starburst amacrine cells shown in black here, in this case 24 of them, and now looked at their contact sites. And that was very interesting because we could see that the situations where amacrine cells, these starburst amacrine cells, make contacts with the ganglion cell were very conspicuous because the presynaptic cells were like wrapped around the postsynaptic dendrite. We confirmed that in data where we had also vesicle staining in there, so we could like really tell what is a synapse and what not. So basically this situation where the wrapping is what was a synapse. In other cases, two neurites just passed and that was not a synapse. And I should mention that in some cases, between two neurites, we found very thin sheets of glia. And this is really a challenge because these are situations where two neurites get as close as easily like 100 nanometers, but instead of actually touching and potentially making a synapse, we saw glia in there. So that means that methods that do not have that kind of resolution can be challenged by that situation, where almost it seems like glia prevents neurites from touching also. So that can.
So a quick question. From time to time, there's been debate about whether you can identify inhibitory versus excitatory synapses based purely on anatomy.
So in the retina, it's particularly difficult because you have dendritic release, you have multiple kinds of transmitters per cell. Starburst amacrine cell is an example. It has both a GABA and acetylcholine release. So in the retina, it's extremely difficult unless you have basically the cell reconstructed and you have other knowledge about it.
You really have, all you can say is there is a synapse here, you can't tell everything.
Yeah, and what you can do is by the type of the cell, which is, however, a morphological identification by the reconstruction, you can link it back to the literature where using other methods, transmitter content has been established. Now, that is retina and comparably kind of variable structures, at least synaptically. In cortex, this has been studied and is being restudied right now by those groups that work on it. It seems like there is a more clear distinction. The classical distinction was that between a GABAergic and a glutamatergic synapse, one being what was called symmetric and having other kinds of vesicle shapes than what is called the asymmetric synapse. It's pretty likely that it is, the actual differences are partly staining artifacts or fixation artifacts, but it seems like they're reasonably consistent, which means you could still use it. Now, having said that, in the data blocks we have, as soon as you have hundreds of microns, really the strongest constraint is knowing about the type of neuron by its morphology. You don't actually have to classify the single synapse anymore, or if so, you can do it for hundreds of them from the same neuron. Yeah. Yeah, so it's really, you're using the massiveness of the data to get rid of that problem. Okay, so we identified synapses, and just to finish up this story very quickly, the question was now whether, in fact, the presynaptic inhibitory neurons would specifically select postsynaptic ganglion cells by the directionality of their dendrites. So remember I had shown you that the starburst amacrine cells, which are shown here in black, react specifically to outward moving light stimuli and do not so much to inward. That means that given the direction of the dendrite, you can predict what direction of motion will excite the starburst cell. Okay? So, and the idea was that if really this circuit is specific and that the presynaptic cell conveys the directionality signal to the postsynaptic cell, then those synapse distributions should respect this directionality of the presynaptic cell. And as you can see here for 2 single-cell examples, and more so even in the population data, we found, in fact, that the type of the postsynaptic neuron determined which part of the amacrine cell wanted to connect. So in other words, the colors of these synapses are the functional properties of the postsynaptic cells. Yeah? So all the black dots are synapses. The colored ones are those made with one of our recorded ganglion cells. And we can see that those starburst amacrine cells specifically chose functionally homogeneous postsynaptic neurons given the directionality of the postsynaptic neurons. So this was a very strong indication already that there's a high specificity in the circuit. It becomes even more apparent if you look from the other side, which is ask for a given postsynaptic cell, again, directional selective ganglion cell, is the input dendrite that comes from the starburst cell respecting my directional preference. And this is summarized here in this polar plot where in color you have the well-known tuning curve that comes from the functional recording, where you plot over directionality of stimulus, you plot the strength of the response along the radial axis, and these kind of tuning curves are well known, and again in color it's the functional one. And what we were able to add to that is the structural tuning curve, shown in black here, which is the distribution of directionality of the dendrites that target the postsynaptic cell. And as you can see for this example, it's nicely visible that actually the input kind of was almost exactly 180 degrees opposite to the functional response. And that opposite direction makes sense because the synapse is inhibitory. So you can, using that data, you can now argue, and this was true for all the cells that we tested, you can now invert the argument and really say it is very likely that the directional preference of the presynaptic inhibitory neuron is directly conveyed to the postsynaptic neuron, and this makes up the directional selectivity of that particular ganglion cell. And this was really exciting and also pretty amazing, because this really means that a single dendrite can very, very selectively decide who to contact. And it's, I think, a nice example of a very specific circuit, which may be true in many cases in our nervous systems, where wiring can be very specific and not just average connectivity based on who can potentially contact who. Here, really, single dendrites decided, apparently, who to contact and made up the functional property of the postsynaptic cell. Okay, so that was that particular story, and just to give you the most recent update, I want to also show our dense reconstruction efforts that we've been pursuing over the last years. This is the last few minutes. So we took a block of tissue again from the retina and tried to get at all of the neurons. So this is the block of tissue. It's roughly 80 times 120 microns in the plane of the retina and spanning almost the entire depth, all the way from the photoreceptors here to the ganglion cell layer there. So this is again just a movie of that tissue block. And then the first thing we did was we just reconstructed all the cell bodies, which you can see here. So these are all the ganglion cell layer cell bodies. These in blue are the amacrine cells, red bipolar cells, and out here the photoreceptors. Okay, these are just cell bodies, not so interesting. But then we asked our tracing army to convert this into reconstructions. So we now have close to 200 students who do this, right? Who like to reconstruct nerve cells and maybe enhanced by our scientific ambitions and the money we pay them, and do this, right? Convert the data set into reconstructions. So this is just a reconstruction of all the bipolar cells in the volume, 460 of them. So these neurons here belong to the cell bodies shown in red. And just to get in, the color code is at this stage was a here actually is a type code now for the bipolar cells here, relating back to the basic on-off and then additional type classification. So this walks you through basically the different types of bipolar cells. It is known that this inner plexiform layer is highly laminated, as you can see here if you walk through, right? So it's a very kind of sheet-like structure when it comes to the projection planes, and that had been noted before. The bipolar cells themselves make up roughly 5 layers of this IPL, or divide the inner plexum layer into 5 layers. This is just a kind of reasonably insight-free propaganda movie to, but I still show it because it kind of gives you an idea of what we have and what we, but also you can see that first of all it's skeleton reconstructions, right, that the humans provided, and there's still a lot of free space which is covered by all the other neurons that we also went for. Okay, so what now? Well, we also wanted to have the ganglion cells because these are the postsynaptic partners. We've reconstructed all of those that were with their soma in the volume. Here, for instance, again, the bistratified one, and again, other types that have and have not been described. There's not only these cells, there's only amacrine cells. We have also reconstructed those. And just want to show you a few more specific examples for the cell types so that you have an idea of where we are. So again, this is now the view we are going to take from now on. Light comes in from here. This is the inner nuclear layer with all the cell bodies. These are the ganglion cells that then send their signals to the brain. And as you can see, right, so as you know, the light goes through all of your retina before it hits the photoreceptors, and then processing happens in this direction, and then ganglion cells to brain. Which part of the brain is the main recipient of the retina? Mitya introduced that today. But the direct next step after the ganglion cells? Thalamus, yeah, and yeah, LGN is the main projection target, at least in mammals like us. And then V1 is the next step. So it's actually, it's 4 synapses from the photoreceptor to the cortex. And that's true for most sensory systems, right? Four synapses to the cortex. Okay, so to link it back to types that have been described before, we looked for those classifications like the one proposed by Wessel, 10 bipolar cell types. We found, of course, rod bipolars, type 9, and so on. So we have them all just to get an idea of the mosaic also. So this is an example of how the dendrites and also the axons of, in this case, type 7 bipolar cells nicely kind of tile the visual space. So each of them basically receives inputs from only one piece of the visual space. And also for ganglion cells, we found many described types. Recently, these genetic classifications, for instance, the W3 as proposed by Sainz and Meister, or the JMB ganglion cells. So this is more for the retina aficionados that this is, we have all those kind of types. Well, was there something interesting when it comes to types already? Well, we also found one that had been kind of suggested may be there, but wasn't really clear and didn't show up in the classifications. We could show that it really is there. It's very sparse. It's a large bipolar cell type that does exist in mouse, a giant bipolar cell, just in the on-off band. So a very interesting one, very sparse, very rare. But if you have hundreds of cells, then you can find the sparse types too. And then we also reconstructed now all the amacrine cells. There are many small field amacrine cells, different stratifications. It's just an overview again. For those who are, you know, yeah. So there's just many types. So as you can see, there's just many different types of cells. Roughly 40-ish amacrine cells, 10-ish bipolar cells, and more like 20-ish ganglion cells. Okay, so that's just the single cells. Of course, more importantly, we wanted to go for contact or connectivity matrices, or at least attempt that. For that, now we combined skeleton tracing and computer reconstruction, looked for touches between neurons. So we converted this kind of gray value data into classifiers. This is work together with Sebastian Sung's group at MIT, who trained neuronal networks, convolutional neuronal networks, to do this classification. Then we developed some segmentation algorithm that is reasonably stable, so it doesn't give us too much under-segmentation. And then we can combine that given some merging rules, so whatever. So in the end, we come up with a segmentation that is reasonably okay locally, that we can then combine with the skeletons in the following way. So we take a skeleton, we overlay the objects from the computer analysis, combine it into one object, and then that is a bipolar cell axon in this case. And then we ask for those cases where neurons touch and do so several times between pairs of neurons. Here is an example where there are 2 conspicuous touches between a bipolar cell terminal and a ganglion cell dendrite, and then we analyze for each of those locations the contact faces and measure the properties of these contact faces to detect what is likely to be a synapse. So there's just one example. And then we come up with what we like a lot because it's a connectivity matrix between, it's more like 900 neurons in the retina. Color here indicates the warmer, the stronger, the and more and larger touches. And these are all of them just listed in the way that we wanted it to be in the beginning. So basically, this is a attempt at a connectivity matrix in the retina of a reasonable size to then analyze and look for interesting features. And just to give an idea of what this means, so each of the entries there corresponds to a pair of cells. So each dot is a pair of cells, and again, the color indicates how strongly those two cells contact each other, both in terms of the number and also size and, of these contacts. And then there are other examples where cells overlap a lot but do not contact, which then would show up as a 0 entry in the matrix. And now, just to get a feel for what we can do with this, I want to give you an example of analysis. One is we asked whether certain types of bipolar cells that are lumped together currently actually can be distinguished by connectivity. So we took a piece of the matrix where the x-axis are bipolar cells and the y-axis are different kinds of ganglion cells. So this is a pre-post matrix, bipolar to ganglion cell matrix, and asked whether simple clustering by innervation allows us to pick out subtypes. And we were just by some hierarchical clustering thought that there may be some pattern here, and then looked more closely, and these were two types of ganglion cells which have and have not been described before. And by then analyzing the kind of overlap probabilities, and then only using those that can potentially innervate the same ganglion cells, which are shown here. So this is now all the bipolar cells that could innervate those two cell types. And by innervation, we can pick them apart and basically find two subtypes, of which one seems to be a reasonable mosaic. The other seems to contain even more types. It's definitely a factor of 2 more than needed. So we're looking now into how we can pick apart those, also probably by American cell innervation. So this is just an example, and really, I think it's really only interesting really for retina, but more conceptually, I think it's interesting because it shows you how you can use connectivity matrix to find structure in the wiring and use that actually to determine what you call a cell type.
Moritz, missed how you went from the touches to synapses, because you said you analyzed the properties of the touch to determine that whether it's a synapse or not, but where do you get ground truth for that?
Mm-hmm. So first of all, what we did is we just, for this particular matrix, report the total contact area, that is number of contacts times average contact area. Now, that is a very strong predictor in the retina, because in the retina, if two cells contact, they do so typically at 5 to 15 locations and not at 1. So the statistics are very strong that if you have many very large contacts, that is very likely to be a connection between these two cells. So that is, there the statistics of number of contacts help you a lot, because one accidental touch is fine, but 15 large ones is just very unlikely. So that is just the way we did it. Now, what is our ground truth? We have a data set that I usually, if there's time, play around a little bit with in the class, where we have everything stained, vesicles, conventional stained data set, and there we've reconstructed some 250 or 300 of these dyadic synapses, and have seen a very strong size correlation. It is not all or nothing, but it is strong enough to convert at least a cell pair that has more than 5 contacts into a very likely synaptic connection. So that is the ground truth we have.
But if you apply this method to the problem of directional selectivity for this starburst endocrine cells to directional selective ganglion cells, would you get the result that you got with Kevin of the specificity of those connections? Just from the area.
Yeah. So their area is also already a strong indicator. We haven't exactly tested for that. That's a good thing we should do. There, actually, what helped on top was the wrapping of the. So it's the geometric feature of the contact. So in that setting, for that particular connection of starburst endocrine cells and ganglion cells, the geometric feature was another strong indicator, which we haven't used here in this matrix, but which of course you could use if you wanted to. Now, I should say one thing though. The analysis I'm showing here right now of this matrix is only based on cell-to-cell connectivity. So we're, in this case, not analyzing single contacts or subcellular specificity. Here, we are so far only analyzing the cell-to-cell connectivity matrix. And I'm trying to show what you can do based on a cell-to-cell matrix. And for that, you do not have to have the single contact certainty, so wherever you want to have that from. For that argument, it's enough to have that statistical argument, which we derive from that other data set. Now, I'm pretty confident that if you take a particular type of connection, you can also find geometrical indicators. But for the matrix I'm showing, we're reasonably confident because it's a cell-to-cell connectivity matrix.
So when two of these arbors are overlapping, how likely is it that they have the synaptic contact?
So there is a strong
That's what people call Peters.
Yeah, exactly. So the Peters, I don't have it here. So the Peters matrix, the rough structure of the Peters matrix is the same. And that is clear in a laminated structure. If you have two cells where one laminates here, the other there, there's no way they can contact, and that is visible by Peters already. However, the more precise structure is not predicted by Peters enough. Especially, and examples for that, I showed this one example, and the more kind of general example is this type 5 bipolar cell, which has a completely common stratification, but we think now we can pull apart 3 different innervation types, and they have the same lamination. So, okay, another point you could make is...
How often is it violated?
The type 5 bipolar cells are like a fifth of the IPL, and all of, so in that sense, that is a very strong example. We haven't, we're looking into type 3. I think there's other examples there. So I think it's the overall, again, in the laminated structure, the overall connectivity is correctly predicted, but the specific connectivity is not. Now, just before your question, one has to say that Peters, for Peters, you have to specify the spatial scale of Peters if you want, right? So you could say, if you do a nanometer Peters, then that is basically what we're doing, right? So in that sense, if you're talking about a light microscopic Peters, where it's usually half to 1 micron or so, that you can do maybe even less, depends on the method, then that's the argument I'm making. So if you had a nanometer Peters, which, yeah, that's...
So would it be possible to give a quantitative comparison, right? So suppose there is this actual connectivity matrix that you establish by looking at the alpha structure and identifying each synapse. Then there is your method, which takes the area of all contacts. And then there is the generalized Peters rule, which looks at, proximities up to a certain range. And can you tell quantitatively how much information is gained by just applying Peters rule on a certain scale, how much more information is gained by using the area of contact versus the specific connectivity that presumably can...
Sure. So for examples, we can do that for, to do it for, we can bootstrap that question. We can, if we wanted to do it exhaustively, we would have to reconstruct another complete data set to do one-to-one. Which is going to happen, but for this particular point, I think it would have to be bootstrapped. I think it's definitely possible, and we've compiled the Peters matrices for different length scales, and the outcome was qualitatively what I just said, that the rough structure is there, that's clear, but the more specific structure is not. So, but that was for a 1.5, I think, micron Peters. So it depends. Then one still has to ask how are you going to get such a Peters matrix? But I think that is a very fair comparison to make, to say, like, what is the scale at which you have to...
Not so much about statistical inference that I have in mind, but one of the developmental strategy system uses to form these connections.
Yeah.
So how often is it enough to just bring the processes close enough, and how often do you actually have that chemical?
Yeah. Okay, so how often is, that's something that one could look into here.
Just speculating about how the circuit develops.
Yeah, the example I gave with the South American cells clearly is one that has to be extremely highly specific because they're very strongly co-laminated. Yeah, yeah, and apparently per dendrite, not per cell. That is amazing. That was the question.
Yes, if you didn't know that one of the ganglion cells are responding to directional motion. Do you think you'd be able to predict that it should respond to directional motion from your reconstructions? And if you could you then predict all the visual primitives that are being computed in the system?
I mean, that is would be a very ambitious undertaking, but I think that in principle, it's not unthinkable to start doing things like that. Now, how is that possible? The question is how much spatial, how spatial is the processing? And in the retina, of course, it is very much so. So you could imagine if you had, like, say, asymmetries in the strength of bipolar cell input on one side, whereas the other of the ganglion cell, that already would make up some kind of direction, or could make up some kind of directionality. So things like that are possible. For the example of a starburst amacrine cell, you have to know that the cell has a certain functional response before you can use that to infer the postsynaptic cell's properties. More generally, I mean, what we can pull out properties like the number of bipolar cell channels a given ganglion cell integrates, for instance. So that will tell you something about the variety of the signal it carries, or the exclusivity for a given ganglion cell compared to other ganglion cells of comparable or that lie at the same location. Properties like that. The specific spatial, yeah, again, so to really get at all the primitives, we need, especially for the amacrine cells, I think we need to know enough about their functional properties to do that exhaustively. That's my suspicion.
Slightly off the topic, but there's been this raging debate for a while about exactly where the directed silicon originates. Is it at the level of the amacrine cells, or at the level of the ganglion cells, or both? That is resolved now?
So, I mean, we think, at least that's how it, I think, was mostly perceived, that this data now strongly made the case for the amacrine cell lending that to the ganglion cell. I don't think this excludes that on top this signal is kind of enhanced by anti-sign excitation or same sign inhibition on top of it.
But the ganglion cells couldn't be doing it independently in parallel through another pathway.
Well, yeah, I mean, that's more difficult to exclude, although we, with that kind of data, can at least exclude strong bipolar cell asymmetry. So we can basically now start to speculate about other possible pathways, and because at least locally the reconstruction is dense, at some point you have to give up and say, okay, if none of the proposed models is there, then probably that's the only one that remains.
And do you see asymmetry in the bipolar cell input onto starburst amacrine cells?
Okay, onto starburst, I looked at DS ganglion cells. I haven't looked at starburst. So no for the DS, but starburst, I haven't really looked at it. But again, these are examples, I mean, these are questions, of course, that can all be asked to that kind of data.
So the retina, I don't, the avian retina at least, I don't know about the mouse, it's infamous for the number of peptides it contains and other modulators. And presumably the mouse has the same kind of attributes. How are you going to work that into the final, any portion of your functional anatomical reconstructions?
So I think that the retina is really an example where synaptic types are very variable, as we mentioned already, and you're absolutely right about that. Many transmitters co-release of multiple transmitters at the same site, and so on. So I think that is, I mean, we don't have that data in that data set. It doesn't tell us that at all. There is luckily a large literature out there pharmacologically and also immuno staining that we can map now nicely onto this, because a lot is related to lamination and morphological properties of cells. That is how I think it will have to be. At the EM level, to have a very strong transmitter marker is, I mean, yes, people are working on that. So I don't think it's unthinkable in the future to have a data set where also transmitter types are labeled. But we don't have that here right now, and right now it's a correlative, or it's adding it from the literature. I think there are other settings like cortex, which I'm actually even more interested in, where transmitter types is not such a big problem because most is glutamate and GABA. So I think it's a specific problem. Just last example. So I told you we found this giant bipolar cell that is in there, and then we can look for the kind of synaptic connectivity of that bipolar cell, just as an example. So we find a very specifically co-laminating amacrine cell that takes up this very, very strong selectivity in the lamination in white bipolar cells, in red and a specific amacrine cell. And then we can go further and ask, so by the way, this is an example of what I mean by many inputs. So if you see a pattern like that where there's a very sparse or reasonably sparse amacrine cell here in the red, but wherever it has a chance, for each of the bipolar cells makes multiple reasonably large contacts, that at least from the other data set what we see, that is very likely to be an innovation by that type of cell to that particular cell. While if there's just one of those small contacts or so, that is much less likely. So that is the level of or like qualitative impression of that statistics. And then we can walk on and ask what is the postsynaptic ganglion cell for that specific amacrine cell, and we can look for a strong innovation there, and then take this ganglion cell. Again, this shows here how this particular amacrine cell in black really makes sure to make a contact with that ganglion cell wherever it can here along the dendrite. And then we can ask what is the bipolar cell input to that ganglion cell, and we find that this is a type 5, part of type 5 bipolar cells that makes this contact, again showing these massive innervations here that we know or think is very likely to be synaptic. And that is just an example of how using this kind of data, we kind of post hoc can do many experiments, right, in a way. So we have a cell type, this bipolar cell that we didn't or weren't aware of before really, and now we see it and we can ask what is actually the wiring of that cell and go step by step and don't have to do an experiment again, but can like really walk through the matrix. And there's another interesting circuit there involved by bistratified amacrine cell that you see in red here, an on-off bistratified amacrine cell, a very rare thing, and that is also involved in that circuit. We are speculating about the function of that wide field bipolar cell based on its photoreceptor contacting. Just again, an example. So find a cell, and we have 2 proposed circuits now that this cell is involved in. And I think this is an example of how structural data can show you very sparse but potentially very interesting circuits, right? So I tried to make this argument in the beginning, and this, again, is a bipolar cell. It was 8 out of 460 cells, so a very rare cell type. But with this kind of data, with structural data, you can now very specifically try to untangle your circuitry and write down proposed circuits that, of course, then can be functionally tested, but that kind of gives you an idea of the hypothesis space of the, or actually cuts down and tells you about the some existing circuits. So to summarize, I try to introduce or talk about this topic of structural neurobiology or connectomics, if you want. Focus on tools first, show you the tools we are using and others are using. Try to make the point that for reconstruction we are heavily relying on combination of crowdsourcing and machine learning right now, and then try to illustrate how we think we can now use this data to start to learn to mine those kind of matrices of whatever certainty. I mean, of course, it's getting more and more certain, the entries in such a matrix, but we're, I think, there to start to map circuits at that scale. I want to thank the people involved. This is work done back then with Winfried Denk and Kevin Brigman in Heidelberg. Kevin is now at the NIH, partly collaborating with Sebastian at MIT. And yeah, of course, the tracing army, of course, is also very, very important. Just very briefly, this is retina. What I'm really excited about, I mentioned this several times, is cortex, because this is where we really do the most interesting computations, I think. And this is what we're currently working on in my group in Munich. So we're trying to assemble the data for 4,000 cell connectivity matrix, which is 1 barrel. So 1 barrel, this is somatosensory cortex of the mouse, contains 4,000 neurons largely connected to each other. And the question is very simply, what kinds of circuit types can we find there? And any of the proposed or completely unknown ones. And I should say for that, we're also using data that is conventionally stained, so the synapse detection is not a problem anymore. So we just have the information about the synapses there. So yeah, it's a very interesting endeavor. So thank you for your attention now. And I think we can now yeah, turn to questions. Oh yeah, I have a little bit of data playing. If I still have time, we can do a little bit of
What's the time constraint on that?
Well, we can stay as long as we want.
Should we
So all I will have is not, it's absolutely not, I'm sorry about the, just want to show you the data, but I have some data here and we could just play a little bit with it, and
How much time?
more like 15 minutes or so.
Should, there's two possibilities. One is we can do that now, or we can do my talk, which will be significantly less than 1.5 hours, and then we could come back and do that more open-ended, so you don't have time pressure.
Whatever.